no fixed ways of going about this, but there are practical microbiological ways of best approaching sampling, handling, storage, shipment. The toxins are hazardous substances. I'll say this again. Somebody asked me, well, how toxic are they? So maybe this is a good point to make. I think it all the time, but I realize that, so how much does it take to kill me, does it? If the EPA, US EPA used to rank things, like ricin and botulism toxin and things like that. And they went from, almost non-toxic to super toxic. All of these cyanobacteria toxins would be ranked in that as super toxins. That is, they are all biologically active lethally at micrograms per kilogram level. Kilogram is 2.2 pounds. A microgram is a millionth of a gram. Bottom line is a very, very small amount. To give you an idea, this bloom that's being lifted in somebody's hand here probably contains several grams of dry weight per liter. If you collected a liter of that thick, heavy bloom would be several grams per liter if you were to dry down a liter. A gram of dry weight of microcystis with a decent level of microcystin in it would be producing 1 to 2 to 3, maybe even 4 or 5 milligrams of toxin per gram dry weight of cells. If the lethal toxicity is 50 micrograms per kilogram and you weigh, say, 50 kilograms, just, the math. So a milligram or two of toxin would be enough to kill you if you ingested it, which would be present in a couple of grams of cells, which would be not a heavy bloom. So when the World Health Organization, the US EPA, sets guidelines and says 1 part per billion is acceptable in a drinking water supply based on a person drinking 2 liters a day, then presumably that 1 microgram or the 2 micrograms, if you drank 2 liters, would show you absolutely no toxic effect. But if that was 2 milligrams in those 2 liters, you'd be dead, okay? So so you can understand then why an animal, a cow or a sheep or something that goes down and drinks a liter or 2 of water, a quart or 2 of water, which cows can drink a couple of gallons or more of water, could easily contain more than a lethal dose just in that concentrated bloom. So freshwater algae don't bioconcentrate like marine toxins into shellfish and fish, but by their biomass accumulation can be concentrated enough to provide a lethal dose. So does that kind of put that in perspective for you? Okay. So let's look then at how we might approach sampling. The goal of sampling is really to obtain a representative example of a water bloom event that can be used for identification, enumeration, and perhaps cyanotoxin analysis. Identification and enumeration are kind of lumped together, the amounts that you would need and the approach that you would need. If you're going to go for cyanotoxin analysis, you have to approach it slightly different, but it's the same water supply. You want to use that information to make decisions regarding potential or observed harmful effects from the presence of those toxigenic cyanobacteria in water supplies. And when you set about designing a sampling monitoring program, my sort of basic statement is that sampling and analysis should be based on project goals, not goals based on sampling. And so don't go out and sample and then set your goals. Set your goals and then go out and sample. And that's fairly easy because you're often driven by budget. And so you better sit down and plan your program. Can you afford 10 samples or can you afford 500 samples in a monitoring season kind of thing? Okay. The US EPA is not very clear on how you might sample. If you go to their website and click sampling, this is what you get. they're pretty vague. It says, it's important to collect samples that reflect the actual site or source conditions and that are handled properly to ensure reliable results. So that leaves you a lot of room to set your own, I'm sure. And that's okay, because I really think that, sure, you need to get started with some basic ideas, but when it really comes down to it, you're driven by your circumstance, what are your tasks, what are you challenged to do, what are you charged to do by your agency, and what is your budget. The only other approach, when it comes to cyanotoxin analysis, when you're collecting for identification and enumeration, we'll mention this again, you don't want to freeze the sample, okay? Because you lyse the cells and you won't be able to do identification, you won't be able to do enumeration if that's what you're tasked to do. However, if you want to preserve the sample for toxin analysis and get a more appropriate measure of toxin, keep in mind that many of these toxins are held inside the cell. And if you sample and only do your analysis on the water, you're going to miss most of the toxin. This is particularly true for microcystin. So, lysing the cells by freezing and thawing or doing some other method of lysis is important for the toxin analysis. So that, those are the kinds of things that the EPA website will alert you to. But beyond that, it's kind of, what's your experience? So I'm giving you my experience when we talk about the rest of this. And it doesn't mean that this isn't what lots of the states do, which is certainly what they do. So your sampling considerations are really location and use of the water body, whether it's a drinking water supply, a recreational water supply, or whether it's a stock water supply. That's your first question. It doesn't help you define the goals. And then the goal of that sampling effort, whether you're doing identification, whether you're doing enumeration, or whether you're doing both. And maybe you're after chlorophyll as an indicator of biomass. And do we need to do cyanotoxin analysis, or at least testing? Of course, testing and analysis would be two different things. You can test, and is it present or absent? But if you want the analysis, do you want to know which one it is and how much is there? Then, of course, that's a separate. The ELISAs, the immunoassays that are prevalent and popular, do a bit of both. They identify, they won't tell you which microcystin it is, but they'll tell you microcystin is there and they'll tell you a level that it's at. And then you can compare that level against guidelines that you might have for, is this a problem or not? But if you want to know which microsystems, then you'll have to go to more elaborate chemical analysis. So some typical locations of blooms in thermally stratified shallow lakes or reservoirs that you want to sample. And I've sampled all of these. You may not have time or money or personnel to sample all of these, but they would certainly include shoreline scums, plankton scums in the open water, scums in or on the sediments, dispersed populations within the epilimnion. Epilimnion is surface down to the thermal layer. Homogeneous populations of phytoplankton dispersed throughout the entire water column during well-mixed, non-stratified conditions. Scums under the ice, and I've seen some very nice examples of toxic blooms frozen in the ice, and they produce lots of toxin. Subsurface or metalimnetic phytoplankton maxima. So these are all the different kinds of, some of the major areas that you have to decide what. And most often you're just, okay, I'm down at the shoreline, it's a swim area, there are animal access, this is where the people bathing, swimming, so certainly I'm going to do that scum. And I'll get to what is slightly better than just grabbing a shoreline scum in a second. Recommendations for location. If this is a drinking water supply, then obviously there's going to be a location in that drinking water source where they're taking water out. They're extracting water for the water treatment plant. So I always recommend 4 samples. You go on a surface in the middle of the water body, and often you'll want to take that in the area where the intake is at some depth, whatever that depth is. Then you want to take a sample at the intake itself, at that intake point in the lake or reservoir. Then you want to sample mid-process stream to see how your water treatment plant is operating, is it working well or not. And then obviously you want to do the finished water. So those are, working with drinking water, this is what was recommended. And I did a study with AWWA Research Foundation 20 years ago. We had 50 water utilities, and this was what I recommended. So we had 50 water utilities sending me samples from these 4 locations on a regular basis. Well, slightly different with recreational water. Heavy and light bloom area at each occupied beach. Some people like to swim in or are exposed to heavy blooms, some to lighter blooms. So you want to have 2 measurements of cell number and maybe toxin content. And then now, more in the open water, where more of the water skiers and jet boats and people fishing or swimming out in the lake. So those are, the 2 typical places. And then the stock water supply, obviously, is just wherever those animals are drinking water from. So those would be just some general recommendations then for sample locations. This is just a picture. This is, what a sampling boat looks like. In this case, this was a project that we had in the St. John River in Florida. But you've got your monitoring probes and your sample containers and your sample bins and so on and so forth. And you're supposed to be wearing a life vest, by the way. Sample collection. I never did used to wear gloves, but now that I'm older I do. But you should always wear gloves. Some say you should wear masks. I don't necessarily think that's required, but if you were collecting samples at the beach in Florida and you had a red tide and you had spray, you'd be wearing a mask and probably goggles as well. But for lake water sampling or reservoir sampling, it's not necessary. And this is just for fun. Obviously, immersion is not recommended unless you've got a graduate student that you want to use as a bioassay. But 2 things. Sample volumes vary. It's really only needed 10 to 20 mils for identification or enumeration. You don't need very much. Unless you plan on archiving the sample, splitting the sample, and sending it to various places and having to store it for long term, you don't need more than 10 to 15 mils to get all the information you need for identification and enumeration. And if it's a heavy bloom, you really don't need much more than that for toxin analysis too. If it's a heavy bloom like, collected here or that very first slide, 100 to 200 mils is plenty to do a toxin analysis. But obviously, if this is a finished water supply, you're going to need about a liter. But you never need more than about a liter or 2 to do any analysis in any water supply. So a few mils to a liter to 2 allows you the full range of whatever it is you're going to do. Containers, drinking water, glass is really the best. There are issues with glass. You send people out in the field with glass, they often come back without glass intact. But if that's the policy, and especially if you're doing bacteriological kinds of tests, you would have amber glass bottles. That's just your standard way of doing microbiological testing. And that's probably a little bit better for the cyanotoxin, cyanobacteria sampling as well, but it's not needed. Plastic is okay. If it is plastic, it should be a high quality plastic like an Algae. An old milk carton or something like that is probably not the best. If you have to store it, you will get leakage of plastic materials into the water sample, which can interfere with toxin analysis. It's not going to interfere with identification or enumeration, but if you want to do toxin analysis, you should be collecting at least in plastic of a high quality. For recreational waters, I would recommend the same approach, just because you want to standardize it. And also if you're doing a livestock water supply. So the kits that I put together for our study with the water utilities a number of years ago were these. Went to Walmart or Target, I don't forget which, and bought out everything they had. And these were big enough to handle 4 bottles. These were 1 liter, 3 1 liters for the 3 samples of finished, midstream, and intake. And then the smaller bottle was the one for the open water, which presumably would have more cells in it. And those would fit nicely in these containers, and then you had a plastic bag with instructions and some gloves and labels and mailing labels and so on and so forth. And you had some freezer bags in the bottom of those containers, and people would always ship them express mail. And you could, of course, recycle those. They came back, you took your samples out, replenished them, sent them back. And so it was an easy way to accomplish the project. Yeah.
So how do you recommend sterilizing bottles for?
I don't sterilize, just make sure they're clean and washed with a detergent and recycled.
For both cyanotoxin
Right.
analysis and also enumeration?
Mm-hmm. Yep. Yeah, sterility is not really an issue.
Your preservation was just ice?
Yeah, blue packs, yeah, again, don't freeze when you're doing identification enumeration. And express mail, they always, they're usually sampled and sent within 48 hours, and you've got them the next day, so that's always adequate. They're fairly, if you collect really thick scums inshore, it's a hot day, you're probably going to get some loss, even in shipping, which comes to another point when I talk about where's the best place to sample. I don't think the best place to sample is the heaviest concentration inshore. So anyway, so that AWAR study is summarized here. This is the sheet that went with each one of those kits, just explaining how to, the different sites and so forth. Yes?
For enumeration, we often preserve with Lugols.
Right.
But you don't recommend that for the?
I do.
Oh, okay.
That's
I'm sorry.
coming on slide, but no, good point. Yeah, well, key point is, what's the best preservative? And we'll talk about that, and Lugols is the answer. So, again, just to point out this intake and intracellular and extracellular toxin, there's quite a bit of variability. Microcystin is mostly retained in the cell, and in fact the research that's done indicates that in fact microcystin is probably a primary metabolite, i.e. the cell's not producing it to kill cows, it's producing it as part of its physiological biochemistry, probably photosynthesis. If you do a localization inside the cells, where is microcystin located? Most of it's located around the photosynthetic membranes in these gram-negative bacteria, which are not chloroplasts, but are thylakoid photosynthetic membranes. And they have a particular function. It just so happens that they also don't like your liver, but it's not why they're being produced. Now, the neurotoxins, maybe, those probably are more directly responsible, say, as an anti-herbivore, because they have more of an activity on invertebrates and things. But that's a different question. So microcystins are retained in the cell. So the question of intracellular, I did that. The question of whether you collect your sample, and if you're doing toxin, you want to get all of the toxin, inside and outside. The microcystin is an example of where this toxin is retained in the cell. Cylindrospermopsin is the opposite. It actively excretes the toxin. So if you collect a bloom that's cylindrospermopsin toxin, 70–80% of it's going to be outside the cell. So if you filter that and just test the cells, you're going to lose a lot of toxin. You have to process the whole sample. So this is an example of the difference. And for a water utility, if they can remove cells, they can keep cells out of the water treatment system through their flocculation, filtration, and they don't stress those cells, then they've removed 90-95% of the toxin right away, which kind of interests me in the case of Lake Erie last year, 2014, why there would have been some breakthrough. They had to have stressed the cells. They had to have treated the cells badly in the water treatment process that causes lysis and allows those toxins to be released. Okay, handling of samples. Collections made with clean containers, amber or clear if dark, or shade, of course, and use gloves. Then the question of preservative. Lugols, 1 mil per 100 mils, or just make it up to the color of a light tea color is kind of the rule of thumb for Lugols to preserve your sample. No preservative if you're going to do toxin analysis. If it's important to preserve, then you really should be collecting 2 samples, one for identification and one for toxin analysis. Handling of toxin-containing cells. We always worked with cells that were dried, and dried powdered cells are extremely hygroscopic. They cling to anything, and they have a charge, and so you always work with them inside a biohazard hood in which there's no positive airflow that could blow those cells out, and then even then wear gloves and a mask. If you're handling water samples where the cells are just in the water, gloves are good enough. And of course, if you spill, clean it up. One thing about the toxins, if you've got a spill on the table or floor or whatever, a Clorox solution is good enough to oxidize toxin in terms of a cleanup. Storage of samples. Here's a little more expansion on the preservation. No perfect preservative. The 3 major ones that you've heard of, in your biology lab, you have formaldehyde. Most people, agencies don't want to handle it anymore, so formaldehyde's not there. It would work, but it doesn't work very well. It distorts cells, bleaches the color. So I don't recommend formaldehyde unless it's all you've got. Likewise, Gluteraldehyde is quite hazardous. It's probably the best preservative because that's what we preserve samples when we prep them for electron microscopy and microscopic analysis. But it's hazardous. Most labs don't want you to use it. So that kind of leaves you with Lugols, the old standard limnological preservative. It stains the starch. It may give you loss of flagella, but we don't worry about that with cyanobacteria. They don't have any appendages. It does tend to wear off over time, so if you're storing samples for a long time, you're going to have to re-Lugols every 6 months to a year. And it can also cause shrinkage, so if you're doing long-term storage and you're worried about biovolumes and things like that, then of course that's not going to work. You're probably going to do the... But Lugols is the best one. So this is an example and it's some summary recommendations from a workshop I did in Ohio in 2011. 1 to 500 ml samples are preferred. Amber bottles are dark plastic. Sample obtained some distance from the scum line. We don't recommend collecting at the scum line. You go out right at the edge of where the visible surface scum is and go about 20 to 30 centimeters below and collect your sample. This gives you a more representative sample of what's really in the water body if you can only sample at the edge. If you have a regular program and you can go everywhere, then you can do these other sample points. But if you're just down there getting one set of samples, wade out to the edge of the bloom, go 20 centimeters below the surface, collect your sample, do your identifications, etc., and you've got a better representation of what is there. Because scums inshore may concentrate different things than are a few yards out too. You're probably going to have better diversity of the organism further out, and some of that diversity may in fact be toxin producers too. Concentrated bloom is okay for toxin analysis if the concentration is calculated based on the biomass. One of the things you do, of course, if you're going to concentrate, do your testing, that's not representative of what the lake is. And you either have to figure out what your concentration factor is, so you can say there's this much in the lake, all you can say is this is what there is in this surface scum inshore. Pencil or waterproof marker, I don't know how many times we've labeled things and you've lost the label. Wear gloves and mask. Keep samples cool and in the shade or the dark is perfectly good while you're sampling. You don't have to worry about getting it to a cold room real quick. Just keep it cool and in the shade. Lugols is representative of the preservatives. And in Ohio's case, this is all up on their website too. Most states now have similar websites for that. Some of those references, determination of cyanobacteria in the laboratory. That's the WHO book that we published in 1999. There is a section in there on sampling, chapter 12. The AWWA has a nice manual. This manual M57 2010 has a whole chapter on sampling and identification. They also have the chapter in there that provides the taxonomy key that I'm going to encourage upon you this afternoon. Standard methods of water and wastewater, the AWWA book. This 21st edition, 2005, there's now a more recent edition, but they have a very extensive section in there on sampling. And the newest edition is going to have a complete chapter by Anne St. Amand, who runs one of the major ID labs in Wisconsin. And she's going to have a full chapter in there elaborating sampling specifically for cyanobacteria. So if you can afford the new version, which of course maybe they're $300 or $400 now, I don't know, but there will be a specific chapter in there by her. In 2008, the EU, when the EU was formed, they spent a lot of money encouraging countries to work together, and one of the things they did was cyanobacteria. So there's quite a bit of European publication. There's one, 2008, Cyanotoxin sampling, sample processing, and toxin uptake. So that's a nice one as well. The problem is you have to buy that. You can't download it. And then the NOWMS website, the North American Lake Management Society, has a website, and they used to have a nice section on sampling and handling and things like that as well. So there are places out there to go as well. But of course, obviously, you know, the state here and the Water Board is going to tell you what they prefer you to do too. But this is what was out there before. Now, this next list is one that I put up on the cloud for you. I put up the PowerPoint lectures, but I also put up 2 PDFs. Did everybody find those as well? Okay. One of those PDFs is this one, Guidelines for Design and Sampling Toxin, Taste, and Odor in Lakes and Reservoirs. It's by the USGS. Jennifer Graham and Keith Lofton are key people in the USGS who've worked on cyanobacteria HABs for years and years. And Keith actually runs probably the best analytical lab for cyanotoxins in the country. His priority is not to take your samples, but he does have some of the best equipment. But they have published this guideline. It's up at that website, and I presume you downloaded it. And it's quite extensive. In fact, let's get back to that in just a minute. He also has put out, the 2 of them, Keith and Jennifer, an analytical methods detection and impacts on data interpretation. If we wanted to get into a more elaborate discussion of recommended analytical methods, that website, and that's actually a PDF of a PowerPoint presentation. So that's also very useful as well. So I'm just giving you some of the newer stuff that is sort of summarizing things from years and years. But that's another good website and a good presentation. If you read this, I can't from up here. What I wanted to throw up here, this is from that analytical, it's a summation of the methods for analysis and detection of cyanotoxins. So, for example, see he's listed biological assays, neurochemical assays, chromatographic methods, liquid chromatography with mass spectrometry, and which toxins they work better with. And so that's kind of the summary out of that presentation. So if you just wanted to, upgrade yourself and see what there is, and so you've got the lingo. This one is kind of interesting too. I thought about running this, but it's a 9-minute US EPA YouTube video, which for the US EPA, this is really nice. They've done a YouTube 9-minute video demonstrating the ELISA, and everyone probably is either going to have to talk ELISA or consider ELISA, and this does a very nice job. It's called Lab Techniques for Detecting Microcystins Using Enzyme-Linked Immunosorbent Assay. So, if you went to that website, I would highly recommend spending your coffee break looking at that sometime. It actually uses the kit that you use. They're generic, they don't promote a kit, but the kit they're using is from a company called Abraxas, which is in Pennsylvania. And they probably have the widest range of kits and assay methods for cyanotoxins. They've pretty much captured the market. Now, actually our lab developed the ELISA kit, I'm bragging, in the late 89, 90, published it, but we didn't pursue a company. This company's pursued and doing very well at it. So that ELISA little video is probably the best thing I can describe to you as to something about that. So, well, I said that I would catch you back on schedule. So that, and I left a little bit of time because there seems to be questions about sampling, because obviously I've left a lot out. I haven't told you exactly what to do, but I've given you guidelines, and or maybe you have some specific questions that are, that come up relative to your situation. If you want to throw them out, sure.
Yeah. So, given the size of Clear Lake, how many and where?
Yeah, you see, you're never going to get an adequate sample representation. Really, what are your activities? Your beach activities, where people are living, mostly, which around there is they're living all around the lake, as I understand. But,
yeah, 3,700.
Right. So I throw up my hand and say, no, you can't. I mean,
if I take it at person A's house at their beach, then person C just down the road is going to be really, really pissed that I didn't take it at their house.
Sure they are. One nice thing about hypereutrophic lakes, though, is that you're probably going to have almost a monoculture. So if you've got microcystis at one point, you're probably going to have microcystis at the neighbor's point.
Right, but is it all doing the same thing? Because we do have examples where we have taken samples rather close together and got a no detect with one and a detection with the other.
Yeah, see, now that's unusual. I certainly agree with different levels of detection of amounts, but never non and highly positive.
We had a 17,000 parts per billion for microcystin near 100 feet from a drinking water intake, and about 50 feet away it was about 400 parts per billion.
And the cell counts were the same.
We didn't do cell counts, it was just we're focusing on toxin levels.
So I guess if I had to, if I provided an answer, I'd say, well, the biomass must have been significantly less, because it's directly related to biomass to toxin amounts.
But we're also finding no cyanobacteria. We're doing cell ID, and we're not seeing any cyanobacteria in some samples. And when we send it, we go ahead and send it off for cyanotoxin analysis. and we are finding cyanotoxins.
Right.
So we have examples of no bloom evident, but toxin levels, and of course, bloom evident and no toxin levels.
And all of those are possible, and, you know, I've seen it all, and all I can tell you is that, you know, you have to pick and choose. Nothing's going to give you all the answers, and you are going to get variability. You're going to get variability in biomass, you're going to get variability in the types of organisms that are present in the toxins. And of course, if you're only looking for microcystin, maybe you've got anotoxin, or cylindrospermopsin is your issue. So there really is no easy way. And what you kind of do is you kind of go forward. When you, as the problem gets worse, you presumably get more resources and you expand it. And you also, if you have the resources, you start looking for other toxins or other organisms that produce toxins, and then you add those to the list. So it's a guaranteed job. But it's tough. There's no easy. Yeah, sorry.
So I wanted to ask you about the issue about where you suggested samples obtain some distance from the scum line. And one of the things that we've been talking a lot about is when you're talking about kids who are playing on the shore and dogs who are at the shore.
Mm-hmm.
Shouldn't we be sampling at the shore right by the scum because that's what they're exposed to? Is that the question as opposed to what is the ambient water quality of this water body out in the water?
Certainly, right. And again, if it's a beach and that's the concern, then you want to know what that scum is too. But if you are limited and you want one number, it's best to go out a bit, collect your sample. Sometimes your charge is, what's the lake doing? Not what's the swimming beach doing. But certainly if it's a swimming beach and you've got kids swimming, dogs paddling, then you want to know what that scum is too. Sorry, Eric?
Yeah. Graph sample versus integrated depth sample.
Right. I prefer integrated depth samples, but that's done best from a boat, not from the shore. And so anything that you can wade out and get, your best sample is 20 to 30 centimeters down. Integrating a whole meter or so, if that's what you can stand in, is not going to be any better than 20 to 30 centimeters because you're going to have all that mixing. But in the open water, yes, an integrated sample is best. A good example of that is Cylindrospermopsis producing Cylindrospermopsin is not a surface scum forming cyanobacteria. In fact, they tend to sit down right around the thermocline. And so in situations where I've seen that, you've only gotten, go down 1 meter, nothing, 2 meters, nothing, 3 meters, a little bit, 4 meters, all of a sudden you got toxin way off the chart. So there's where your integrated sample works. And maybe that's where your intake is too. Yep.
A lot of the lakes look like it starts on one end. I don't know if it's the wind, direction. There seem to be, is it more depth related or surface activity?
Well, yes. Keep in mind that these organisms are growing from the bottom up, not the top down. And when they're on the surface, they're on their way out. Or they're waiting to be mixed by the next windstorm or rainstorm that comes along. They go up to, they regulate, and cyanobacteria regulate their buoyancy with internal structures called gas vacuoles. And they can osmotically regulate the expansion and collapse of those vacuoles. And they're looking to optimize the light regime. The surface light regime is kind of suicide. It's usually too much, so they want to regulate it down in.
Have you ever looked at time of day?
And time of day is very important, sure, yeah. At night they'll tend to be up, a little more, and then at morning the sun comes out, they'll start to go down. But they're highly influenced by what's the wind doing and what the currents are doing and where the motorboats are and people swimming too. So, but yes, they grow best at some depth where the optimum light is. And They also don't like light conditions that are strong, not like some other algae or plants. Their light tolerance is really quite a bit less than a normal plant. Yeah.
So how long can a molecule of a cyanotoxin be toxic?
Question is, how long do the toxins stay active or toxic in the water supply? It depends a lot on the toxin. So start with the most susceptible. The antitoxins are most susceptible. Antitoxin AS, the cholinesterase phosphate compound, the OP, is very sensitive to light and is easily degraded. Antitoxin's a little more resistant. Even microcystin, one of the main ways microcystin would be degraded is with light oxidation. But keep in mind that if you have a surface scum that comes up, you'll get surface scums that are several inches thick on the shore. Only that top layer is going to be inactivated. You go a centimeter down and the toxins are completely active. Well, the fact that microcystins are resistant toxins to begin with. The cyclic peptide composition makes them resistant to temperature in particular. So you can boil microcystin and it's not inactivated. But bacteria degrade them. If you have a regular bloom like Clear Lake, I'll bet you've got bacteria in there that can degrade microcystin quite easily. So it doesn't take long, opportunistic bacteria.
Are we talking like days or weeks or months?
Well, you've had blooms for a long time, so I'm sure they're there. But yeah, if you had persistent blooms, repeating themselves for a year or two, you'd establish a population of bacteria very quickly.
Right. But
It's not days, it's not even months. But
as far as the longevity of the toxin.
Yeah, and then the toxins that are produced, though, are very stable. So if you had a surface scum and you collected the bottom of that scum, it would be months it's going to be there. Right. But usually winter comes along and brings it all in. But in terms of summer bloom, if you have a thick scum on the shore, the toxin's going to be active for the entire period of the time the scum is there.
And would there be any reason why you would want to, say you've got a 2-acre beach that you want to sample, and you only have money for one sample, would there be any reason why you would want to take a composite sample over those 2 acres of beach?
That's certainly possible if you only have money for one sample.
Right.
Yeah.
So
but you start diminishing return because you need a few mils if you're going to do a toxin analysis. And if you're only collecting 100 mils, you can only do maybe a handful of samples before you collected too little that you didn't know what was there.
Or you could just get a 5-gallon bucket and keep pouring from different areas
And
into a 5-gallon bucket and then
Right.
take from that.
Right, yeah.
Okay.
Yeah, if you're limited in terms of the numbers of samples you can process, then,
Yeah, what about a situation where you're using like an algicide to kill bloom off, and then we've had different people tell us how long the toxins will persist in those conditions?
Okay, if you say you lysed a bloom with copper, do you use copper sulfate in California or not?
I usually pack.
Just pack 27, yeah.
We use copper sulfate.
Yeah, I thought so, but I don't know, but I would think if you lysed the bloom, you would be probably having some toxin measure days to a week or two, but it wouldn't last months. Okay?
Are you going
yeah.
to talk about akinines more later?
What do I know about akinines?
I know very little. I'm under the impression that only certain species generate
Right.
them, and so I'm wondering.
Only filamentous ones, and only ones that also produce heterocysts. So if they have heterocysts, they also produce akinines, or can produce them. So it's only those filamentous ones that are in the nostoc group,
So
nostoc
microcysts Microcystis.
Yeah, none of the colonial unicellular ones would have heterocysts or aconites.
Okay.
Remember, aconites are the spores. They're fascinating, but they're hard to work with. I've collected them out of sediments and tried to germinate them, and you can germinate them and produce the organism, but they don't necessarily tell you that, you know, unless you're good at identifying aconites and know they belong to Andina or, you know, Phanozomenon or something else.
Thank you.
Yes.
So then if you're dealing with an area that doesn't have a species that has the aconites, and then you overwinter, where is the supply coming then from the bloom following the next year?
I'm waiting for that question. Microcystis, of course, where does it go in the wintertime? Microcystis overwinters as a vegetative cell in the sediment. You know, remember your sediments are, they don't freeze, usually 4 degrees, is the maximum they'll get. And so they overwinter. So then it begs the question, why do you need aconites? Well, you really don't, but some produce them. And microcystis is very good at overwintering as a vegetative cell.